Okay, imagine taking something we usually run away from a virus and training it, arming it, and sending it in to fight cancer. Sounds wild, right? But that's the core idea behind oncolytic virotherapy, or OBT.
It really is. And what's truly fascinating is that while the idea itself isn't brand new, it really hit a wall for a while. Back in the 1970s and 80s, progress in OVT had kind of stalled out.
Right.
So our mission in this deep dive is to unpack the incredible story of how two, well, seemingly different fields, molecular biology and immunology, basically gave us the knowledge and the tools needed to pull OVT out of that slump.
And transform it into, well, what it is today, a really dynamic area of cancer research.
Exactly.
We've gone through a ton of articles, research papers, our own notes to piece this whole transformation together for you. So let's get into it. Okay, let's set that stage first, the 70s and 80s. So OVT is kind of treading water.
Yeah, pretty much.
But just outside that field, things were like exploding, right?
In immunology and molecular biology. Absolutely.
Yeah.
Fundamental immunology was taking huge leaps in understanding how the immune system actually works. And at the same time, molecular biology was literally inventing ways to manipulate DNA.
And that parallel growth, you're saying, that's the key to OVT coming back.
It's fundamental. You can't understand modern OVT without it. And the immunology part, well, it really starts with this concept from way back.
Okay.
1957. Lewis Thomas, McFarlane Burnett, they proposed this idea of cancer immunosurveillance.
Immunosurveillance. Like the immune system is patrolling.
Exactly. The idea was your immune system isn't just fighting off colds, it's constantly on the lookout, finding and knocking out early cancer cells before they become a real problem.
Okay.
Now, it was debated, refined over decades, but it planted a crucial seed. The immune system could potentially recognize and fight cancer.
And then we started figuring out who the key players were in that fight.
Right. Identifying T-lymphocytes, especially in the mid to late 60s, was massive, and specifically cytotoxic T-lymphocytes, CTLs.
The immune system's assassins.
Pretty much. They could directly kill target cells, including tumor cells. But, just knowing they exist isn't enough.
You need to work with them.
Exactly. And that's where interleukin-2, or IL-2, comes in. Discovered in 1976.
I've heard of IL-2. That was a big deal.
Huge, because it finally allowed scientists to grow T cells outside the body in the lab. You could culture them, expand them, study them properly.
Which opens the door to thinking about T-cell therapies.
Totally. And IL-2 itself actually became an approved cancer therapy later on.
Okay, and what about interferons? I know they're involved somehow.
Right, IFNs. Discovered even earlier, back in the 50s. They were known to have antiviral effects, but also anti-tumor activity. IFN-alpha became a therapy too.
But what was the specific link to OVT?
The key insight was realizing that a lot of cancer cells have defects in their interferon signaling pathways. Meaning they're worse at defending themselves against viruses compared to normal cells. Their internal alarm system against viruses is broken, essentially.
Okay, so that's a vulnerability, a weakness that an oncolytic virus could potentially exploit.
Precisely. It makes the cancer cells selectively more susceptible to viral infection.
So we have the killer cells, the T cells, ways to grow them, like IL-2, and this vulnerability linked to interferons. But how do the T cells know which cells are cancerous?
Good question. That comes down to identifying the target's tumor-associated antigens, TAAs, and later neoantigens.
So these are like unique flags on the cancer cell.
Exactly. Molecular fingerprints, often mutated proteins that aren't usually found on normal cells. These give the T cells something specific to recognize and attack.
Makes sense. And then came the immune checkpoints. That feels like a really critical piece of the puzzle for modern immunotherapy.
Absolutely a game changer. CTLA-4, identified in 87, then later PD-1, PD-L1. These are basically natural brakes on your immune system.
Brakes you need normally, right, to stop autoimmunity.
You absolutely need them. They prevent the immune system from going overboard and attacking healthy tissue. Vital for self-tolerance.
But in cancer?
In cancer, the tumors can sometimes hijack these checkpoints to put the brakes on the anti-tumor T-cell response.
They use our own safety system against us.
Kinda, yeah. And that's where James Allison's insight in the mid-90s was so brilliant, he thought, what if we block the brakes? Specifically, CTLA-4.
Take the foot off the brake pedal.
Exactly. And his work showed that blocking these checkpoints could unleash a much more powerful T-cell attack against the tumor. This was the foundation for checkpoint inhibitor drugs.
Which have been incredibly successful. But how do you make the drugs? How do you make something specific enough to block just CTLA-4 or PD-1?
Right, you need a way to mass produce highly specific molecules. And that technology arrived thanks to Kohler and Milstein in the 1970s monoclonal antibody technology.
Okay, so that let them make large amounts of identical antibodies targeting one specific thing.
Precisely. It provided the manufacturing power needed for therapies like checkpoint inhibitors.
Okay, so immunology is figuring out the who and how of the immune fight against cancer. The cells, the targets, the breaks. But how do you get a virus, this genetic package, involved? How do you engineer it?
And that's where the other revolution comes in. Molecular biology. Arguably, the recombinant DNA revolution was the single most enabling technology for modern OVT.
Right. This is the early 70s. Berg, Boyer, Cohen.
Those are the pioneers. What they developed was this unbelievable ability to cut, paste, and copy DNA with incredible precision, manipulating the code of life.
So what were the tools they developed?
The core toolkit included things like restriction enzymes. Think of them as molecular scissors that cut DNA only at very specific recognition sequences.
Okay.
Then DNA ligases in the molecular glue to stick DNA fragments back together in new combinations.
Right.
And vectors, things like modified viruses or plasmids, that could carry these new DNA constructs into cells to be copied or cloned and expressed.
So you could isolate genes, change them, combine DNA from different sources.
Wow. It was transformative, so powerful, in fact, that scientists themselves called the Asilomar Conference in 1975 to discuss the potential risks and set up safety guidelines.
That's fascinating. They knew they'd unleash something huge.
They did. And the potential for good was also obvious. Genentech, the first biotech company, was founded in '76, and they quickly started producing things like recombinant human insulin using this tech.
Okay, but back to OVT. What did recombinant DNA mean for viruses?
It meant everything. It provided the means to directly engineer viral genomes. Before this, you were basically stuck screening naturally occurring viruses.
Finding one that just happened to work okay?
Pretty much. Now, you could go in and make specific changes, delete genes that cause disease, making the virus safer, insert new genes, therapeutic genes, modify the virus's surface proteins to change which cells it infects. And crucially, you could finally study precisely what each viral gene actually did. So, fast forward to the late 80s, early 90s, you've got this convergence.
Right, the immunology insights and the molecular biology tools.
Exactly. The understanding of cancer's immune vulnerabilities meets the ability to custom-build viruses. This is what really set the stage for OVT's comeback.
It shifted the whole strategy, didn't it? From just finding viruses to actually designing them.
Absolutely fundamental shift. That first wave of OVT relied on empirical selection. Finding viruses in nature, it was hit or miss.
Inconsistent safety issues.
Big time. Recombinant DNA allowed for rational design. You could use your knowledge of the virus and the cancer cell to modify the virus deliberately.
To make it safer, more effective, more tumor-specific.
Precisely. You could delete virulence genes. You could engineer it to only replicate in tumor cells by exploiting those vulnerabilities we talked about, like broken interferon pathways.
Okay.
And you could start arming the viruses, putting therapeutic genes into the virus.
And we saw proof this could work.
Yes, a landmark study came out in 1991 from Robert Martuse's group. They took herpes simplex virus, HSV1, and deleted a gene called thymidine kinase, PK.
Okay, TK, why that gene?
Well, normal, non-dividing cells, like in the brain, rely heavily on the viral TK for the virus to replicate efficiently. Deleting it really weakened or attenuated the virus in normal cells.
Making it safer.
Right. But many brain tumor cells, gliomas, have high levels of their own cellular TK, enough to compensate for the missing viral gene.
Ah. So the engineered virus could replicate well in the tumor cells, but not so much in the normal brain cells.
Exactly. It showed selective replication. It was a huge proof of principle that you could rationally engineer a virus for improved tumor selectivity and safety. But it's really striking when you look at the timeline of all this, the gap between these fundamental discoveries and actual clinical therapies.
Yeah, I noticed that too. T cells identified in the 60s, but GAR-T therapies and widespread checkpoint use, that's like 2010s onwards.
Right. IL-2 discovered in '76, approved in the '90s. Recombinant DNA in the early '70s. Engineered OVs hitting trials mid-'90s. TVE approved after 2005. There's a significant lag.
So what does that tell us? Why the delay?
Well, it suggests that foundational breakthroughs often need other things to catch up. Maybe complementary technologies like monoclonal antibodies for checkpoint inhibitors.
Or maybe just a deeper understanding of the biology.
Exactly. A better grasp of the tumor microenvironment, for instance. The basic science needs time to mature, and the technology needs to develop alongside it before you get effective therapies.
So OVT is a perfect example. The old idea needed the new molecular biology tools to become truly viable.
Precisely. The engineering capability unlocked the potential that was always conceptually there.
And speaking of potential, there was another shift in thinking around the mid-2000s, wasn't there? About what OVs are really doing.
Yes, a really important evolution. Researchers realized the virus killing tumor cells directly assisting lysis that's important, but it might not even be the main benefit.
Oh, so what else is it doing?
Increasingly, the focus shifted. People started seeing the virus as an in situ vaccine.
In situ, meaning in place. Like it generates the vaccine right there in the tumor.
Exactly like that. As the virus replicates inside the tumor cells and eventually bursts them open, it does two critical things simultaneously. First, it spills the guts of the tumor cell, releasing all those tumor antigens, these wanted flags we talked about.
Making them visible to the immune system.
Right. And second, the viral infection itself and the messy way the cells die creates a lot of danger signals. It causes inflammation, releases molecules that basically shout to the immune system, hey, something's wrong here.
So it's like setting off flares and showing the enemy's picture at the same time.
That's a great analogy. It attracts immune cells to the tumor dendritic cells, T cells, and helps activate them against those newly exposed tumor antigens. The tumor essentially becomes its own vaccine factory.
Wow. Okay, and this explains the whole arming strategy then, putting extra genes into the virus.
Absolutely. This shift towards thinking of OVs as immunomodulators, as immune triggers, is why we now engineer them to carry extra immunostimulatory genes.
Like GM-CSF and TVEC.
Exactly. GM-CSF helps recruit and activate dendritic cells, which are crucial for kicking off the immune response. And it's why combining OVs with other immunotherapies, especially checkpoint inhibitors, is such a hot area.
You get the virus stirring things up, releasing antigens, and then the checkpoint inhibitor takes the brakes off the T cells that come running.
That's the idea, a synergistic effect. It aligns OVT perfectly with the broader goals of modern cancer immunotherapy, using multiple approaches to get a powerful, targeted immune attack.
Okay, so let's get into the nitty-gritty of that engineering, because that's really the heart of modern OVT, right? Making these viruses smart.
It really is. The primary goal is always to maximize the therapeutic window.
Meaning
You want maximum killing and replication of the tumor,
Okay.
minimum harm, minimum toxicity in normal healthy tissues, enhance tumor selectivity, minimize side effects.
And you mentioned two main strategies for selectivity.
Broadly, yes. The first is transcriptional targeting. This controls where the virus replicates by exploiting molecular differences inside the cancer cells compared to normal cells.
Okay, how does that work? Give me an example.
One common method is targeted gene deletions.
Yeah.
You remove specific viral genes that the virus absolutely needs to replicate in a normal cell, but that a cancer cell, because it's already messed up in certain ways, doesn't require the virus to have.
Like the HSV TK example again. Normal brain cells needed the viral TK, but the glioma cells didn't, so deleting it made the virus selective.
Exactly. Another classic example is deleting the adenovirus E1b55K gene. This protein normally deals with the cell's p53 tumor suppressor pathway. Many cancers have mutated, non-functional p53.
Right, p53 is often broken in cancer.
So the virus lacking E1b55K can replicate well in p53 deficient cancer cells, but it's restricted in normal cells with functional p53.
So you're exploiting the cancer's own defects.
Precisely. Or deleting viral genes that fight the interferon response, since many cancer cells have faulty IFN signaling anyway.
The virus doesn't need its anti-IFN tools as much in the tumor, but does need them in normal cells, making it safer in normal tissue.
You got it. Another approach under transcriptional targeting is using tumor-specific promoters.
Like a switch that only turns on in cancer cells.
Basically, yeah.
Yeah.
You put essential viral replication genes under the control of a promoter sequence that's known to be highly active only in certain cancer types, like the PSA promoter for prostate cancer, or HT-ERT, which is active in many cancers. So the virus only really gets going in the target tumor cells.
Clever. And what about that microRNA thing? That sounded complex.
MicroRNA detargeting. It's elegant. You engineer artificial binding sites for specific microRNAs into essential viral genes.
MicroRNAs are tiny RNAs that regulate genes.
Right. Now imagine a specific microRNA is highly abundant in, say, normal liver cells, but very low in liver cancer cells.
Okay.
When the engineered virus enters a normal liver cell, that abundant microRNA binds to the artificial sites on the viral gene and silences it, shuts the virus down. But in the liver tumor cell, where that mirin A is scarce, the viral gene isn't silenced and the virus replicates.
Wow, so you use the cell's own regulatory molecules to control the virus.
Exactly. It allows for very fine-tuned, tissue-specific detargeting.
Okay, so all of that is transcriptional targeting controlling replication inside the cell. What was the other main strategy?
Transductional targeting. This one aims to control which cells the virus can even enter in the first place.
By changing the virus's key.
Sort of. You modify the proteins on the virus's outer surface so that it can only bind to receptors that are specifically overexpressed on cancer cells, trying to make entry itself tumor-specific.
Sounds ideal, but you said it's tricky.
It is. Engineering viral surface proteins without messing up the virus's ability to assemble or infect properly is challenging. Getting high selectivity and high efficiency this way has proven difficult, though people are definitely still working on it, mostly with adenoviruses and HSV.
Okay, so selectivity is key. But then there's the arming part, making the virus do more than just kill cells.
Exactly. This is where genetic engineering really shines. We insert therapeutic transgenes into the virus. The virus becomes a delivery system, producing these beneficial molecules right inside the tumor as it spreads.
And what kind of weapons are they arming these viruses with?
All sorts. A major category is immunostimulatory molecules, cytokines, and chemokines.
Things to rally the immune system.
Right, like GM-CSF to recruit dendritic cells, interleukins like IL-12, IL-15 to boost T cells and NK cells, interferons to enhance antigen presentation, chemokines to physically attract immune cells into the tumor, basically turning a cold tumor microenvironment hot.
So the virus arrives, starts replicating, and starts pumping out these signals like a loudspeaker saying, "Immune system, get over here."
Pretty much. Another really exciting payload is immune checkpoint inhibitors.
Ah, so instead of injecting antibodies systemically,
The virus makes them right there in the tumor. It can be engineered to produce antibodies or fragments against PD-1, PD-L1, CTLA-4.
what's the advantage of making it locally?
Potentially much higher concentrations right where you need it in the tumor microenvironment, with hopefully fewer side effects throughout the rest of the body compared to standard IV infusion. The tumor becomes a local checkpoint blockade factory.
That's very cool. What about those bites you mentioned earlier?
Bispecific T-cell engagers. These are engineered proteins with two ends. One grabs a T-cell, the other grabs a tumor cell.
Like molecular handcuffs linking the killer to the target.
Exactly. It forces them together to trigger killing. Arming an OV with a gene for a bite means these potent engagers are produced right within the tumor, directing T-cells against nearby cancer cells.
And you could even arm them with tumor antigens to boost that vaccine effect.
Yes, absolutely. Engineering OVs to express specific tumor antigens or neoantigens can enhance the priming of tumor-specific T cells, essentially delivering a personalized cancer vaccine directly to the tumor site, along with the inflammatory signals from the virus itself. And there are others too, anti-angiogenic factors to starve the tumor's blood supply, pro-apoptotic proteins to trigger cell suicide, even enzymes that convert a harmless prodrug into a potent toxin only inside the infected tumor. The possibilities are huge.
It really sounds like the strategy has evolved from just, finding a virus that kills cancer to engineering these incredibly complex multitasking machines.
It really has. We've gone from simple gene deletions, like in the early days with Omen YX015, to viruses carrying multiple sophisticated payloads, bites, combinations of cytokines, checkpoint inhibitors. It reflects a much deeper understanding of just how complex the tumor and its environment are.
You need to hit multiple pathways, recruit different parts of the immune system.
Often, yes. The virus has shifted from being just a selective killer to being an active immunomodulator and a really versatile delivery platform for all sorts of therapeutic agents. But of course, there are always trade-offs in the engineering. Choosing between transcriptional targeting or transductional targeting, deciding which payloads to include and how many, it's complex.
Right. Transductional sounds great for specificity, but is technically hard. Transcriptional exploits common cancer weaknesses, but maybe isn't perfectly selective for every single cell.
Exactly. And mirin A targeting offers great control, but depends on knowing the right mirin A profiles. There's no single best engineered virus yet.
So the optimal design depends on the virus, the tumor type.
And exactly what you're trying to achieve therapeutically. It's a very active area of research, constantly refining these strategies.
Wow. Okay, so just to recap this journey we've taken you on, we started with oncolytic virotherapy, this really cool idea that kind of got stuck for a couple of decades.
Yeah, it was stalled, but then it was completely transformed, fundamentally reshaped by breakthroughs in two other fields. The deep insights coming from modern immunology and the sheer power of the tools developed in molecular biology.
And the modern approach, it's this sophisticated blend. You still get the direct tumor killing from the virus, but increasingly it's engineered to be this potent immune trigger.
Actively shaping the immune response within the tumor, all enabled by precise genetic engineering. They're becoming these multifunctional therapeutic factories delivered right to the cancer.
It's just a fantastic example of how breakthroughs in fundamental science, sometimes decades ago, can pave the way for completely new ways to treat diseases like cancer.
Absolutely. That understanding of basic biology is the bedrock.
So, thinking about that time lag we discussed between discovery and clinical use, and seeing how sophisticated these engineered viruses are becoming, acting as these complex local therapeutic platforms, it makes you wonder, doesn't it? What other truly revolutionary ideas, maybe in basic cell biology or epigenetics or RNA biology, are being figured out in labs right now that might be quietly laying the foundation for the next generation of cancer therapies we probably can't even properly imagine yet?